taken malignant cells, T4-2 cells, and what she's done is she's added a drug to accomplish what is called reversion. And so we take exactly the same cells with exactly the same genome, and I add a drug, and I now see that these cells have a much less malignant phenotype. They're more ordered, and the size of the aggregate is smaller, and so forth. And so this is emphasizing something that Valerie has already touched on. It's emphasizing this idea that if you're a cell, just because you have mutations in your genome, it doesn't matter that you're necessarily going to be a cancer cell. That really, that decision to be a cancer cell or to exhibit a cancer phenotype really emerges out of the interplay of the mutations you carry as well as your interactions with your environment. And this is typically, people talk about the microenvironment and so forth. And there are now known a wide variety of compounds you can add to accomplish this reversion process, as well as it's beginning to be understood due to Valerie how shear stresses feed into decisions that cells make to revert or not revert. And so what this is telling us that just because you have mutations in your genome doesn't necessarily mean that you will actually behave like a cancer cell. I'm going to fast forward to this because we're very low on time. So the second general problem in biology, I think has to do with something that we've probably all been fascinated about since we were kids, and looking at a tadpole grow into a frog. How is it that one takes a single cell and over some period of time reliably ends up with something like a frog or one of us? We are a collection of about 10 trillion cells, 10 trillion. It's a very, very large number. Each one of those 10 trillion cells is in a particular state, a neuron, a fibroblast, or something like that. And imagine what has to go into forming you and me or a frog in a controlled manner, in a reliable manner, from one cell to this organism, composed of all these cell types, and typically they're quite stable. Our nose stays a nose, our skin typically stays skin, and so forth. And this is, okay, this is now the full frog, or Xenophysalis. And part of the story, part of the secret for how this process is coordinated involves mechanical forces. And so just imagine I give you a balloon, and you press on the balloon. Then that balloon deforms in a characteristic manner. And wherever in life you see a displacement, a deformation, a warping of something, that's because something else is pushing on it. So whenever you look at a tissue and you see tissue, the cell shape change, or things being squeezed or deforming, imagine a pathologist looking under the microscope, looking at a tissue sample and saying, these cells look funny. They have a different aspect ratio or something. That immediately tells you that the forces acting on the cell must have changed. And so retrospectively, if you think about this this way, then the basic, what a pathologist is looking for also for staging of cancers is they're looking at a snapshot of how the forces changed and how the tissue responded to those changing forces to diagnose and stage cancer. So here's a movie of now a frog forming. And this is not the frog, that was a fly. Here we go. So this is the beginnings of the formation of the nervous system of the frog and all the internal organs. And the tissues, the cells that are going to form the internal organs have now invaginated, and you're beginning to see the nervous system of the frog form. And so every single of these cells in this case is moving. Every single cell is being deformed. And part of the secret for the overall coordination of this process is probably the forces acting on this entire system. And these forces are sensed by all the cells in this viscoelastic, mechanically connected system. And so retrospectively, all the things that Valerie is learning make a lot of sense in this developmental biology context, because cells need to continuously interrogate their mechanical environment, and they're using those cues to figure out where they are in the tissue and what they should be doing. And number 3, explicitly, which is what Valerie has already given you a wonderful introduction on. And this is a picture of cells. And the green stuff is the mechanical spaghetti, the mechanical fibers that cells are using to stabilize themselves. And this fibrous structure, these actin filaments, are mechanically connected to all their neighbors. So if I tug on that cell in the corner, all the other cells will be tugged in response to my perturbation of that cell up there. And of course, if I cut the tissue, then the cells over there no longer feel my mechanical deformation of these tissues here. So as I pull on my skin right now, I'm literally pulling against some of these fibers that are mechanically coupling the cells. And you can imagine all the interesting effects that arise out of this mechanical coupling. And a little bit of history, Valerie again has introduced this. This is an old papyrus, 1600 BCE from Egypt. And this is the first mention of cancer diagnosis based on shape and hardness. And in fact, the words cancer and oncology are based on a particular shape that tumors have and on the swelling associated with tumors. And of course, in those days, the treatment prescription was that this is basically a statement of the god Zenus against you, and therefore you should do nothing against tumors because you would, of course, be acting against god Zenus. Fortunately, we've moved on considerably since those times, and now, of course, we still rely on differential tissue mechanics for diagnosis, and I won't spend any time on this because Valerie has introduced this. And so to summarize the third big mystery in biology, it's this question of integrating genes with mechanical forces. So we know that mechanical forces are important in developmental biology and disease and so forth. We know that genes are important, and we know that signaling networks are important. What we're extremely bad at is combining those 3 things and beginning to understand how those things relate. There are early advances, and here's a beautiful, absolutely beautiful example. The picture on the right is a picture of 4 soap bubbles. The picture on the left is a particular structure formed in the eye of the fruit fly. And you see here, you see 4 cells, and if you just look at how those cells are touching, then that reveals that the forces that control the shape of those cells are exactly the same forces that lead to the same pattern in 4 soap bubbles. And so that tells you that for this particular system, surface tension must be important. And so now you can begin to write statements. And this is one of the first equations, so if you don't remember anything else from today, just remember this one equation. And what's absolutely beautiful about this equation, let me walk you through it. So this symbol is an energy, and so the overall statement is a statement about, in some sense, the total energy of the system. And what's absolutely beautiful about this equation is that this term right here is a deformation. And so this is what you would get, for example, by playing with a rubber band. And so this is a statement about rubber bands. And it says nothing about biology. And these terms right here have gamma sub E terms, gamma sub N. These are cadherins and molecules that Valerie has spoken about. So what this equation does, it combines rubber bands and membranes of drums and soap bubbles with molecules that Valerie has already spoken about. And so this is a snapshot of possibly what we'll be doing a lot more of. And so then one can make specific predictions now based on soap bubbles for the fruit fly eye. One can make mutations, one can start playing with these genetic terms, but also these physical terms, and one can now begin to, in a controlled way, play with the architecture of the fruit fly eye. And so this is an example of one system where we're learning a little bit of what these basic rules are. And to conclude, so I've tried to introduce you to 3 big mysteries in biology that I think all relate to disease in one way or the other. To do that a little bit more explicitly, So in cancer, we're dealing with several hundred different types of cancers, right? Each cancer has several stages.